So we're going to go over chapter 18, and this finishes up the nervous system. You might have noticed that we skipped a few chapters, and we're just covering 13, 14, 16, and 18. So this will finish up the nervous system for us. So every plasma membrane has functions as a receptor for cells, and so there are receptors that are specific for chemical stimuli, electrical stimuli, mechanical stimuli. The fact is that not all cell membranes are the same. Not all cells are the same, and there's this differentiation that we talked about in the beginning. So sensory receptors are very specialized for depending on the area and where they're located and what kind of sensory modality they respond to. So let's define sensory receptor. It is a specialized cell or cell process that monitors either the conditions inside the body or the conditions outside the body. So that would be external environment. Sensory afferent information arrives at the CNS. Well, what does that mean? Well, it has to first be kind of started by some sensation. And so sensory receptors basically respond to specific stimuli, and that stimuli is specific to that specific receptor. That is to say, if you have a touch receptor, you have to actually physically distort or push on that receptor ending to get it to generate an action potential, which would be a sensation, right? So we have stimulation and sensation. So as I say, the stimulation of the baroreceptor or touch receptor. So here's an example. You have a touch receptor and you push on it. So that is physical distortion, and that generates an action potential, which is an electrical current. And that action potential goes to central nervous system. The stimulation is going to be pushing, right? So this is stimulation here, is pushing on the sensory receptor ending. And then the sensation is, oops, did not mean to do that. So the sensation is this action potential being generated and going into the central nervous system. Now, perception is a conscious awareness of this action potential arriving in your cerebrum. So perception is something that, you know, when the sensory information makes it up to your cerebral cortex and your, depending on what kind of sensory input it is, and what cortex it goes to, but it's your conscious awareness of this these action potentials coming into your brain, that is perception. So sensory receptors respond to specific stimuli. If it's in your eyes, you have photoreceptors. They only respond to light waves. If you, in the cochlea, in the inner ear, these little hair receptors really are only going to be bent by sound waves, not the actual sound waves themselves, but sound waves get transduced into mechanical energy, which then moves fluid in the cochlea, which then bends the hair receptors. So my point is, every sensory receptor has a specificity to it. It's specific to a specific sensory modality. And so we classify senses into general senses and special senses. So general senses, we actually refer to these as the somatosensory. Right, and these go to the postcentral gyrus or primary sensory cortex, and some books actually refer to it as the primary somatosensory cortex, which I have written down here. So general senses refer to temperature, those receptors that are monitoring the temperature, pain, touch, pressure, vibration, and proprioception. So this is body position, muscle length, joint angle, and tendon tension. General senses, the sensory receptor is part of the afferent sensory neuron. It's an all-in-one unit. It's more simplistic, and all of these general senses go to the somatosensory cortex, which is the postcentral gyrus. Special senses, on the other hand, are more complex. The receptor that is responsible for being stimulated by whatever type of sensation, whether it's light waves, sound waves, chemicals that you're eating dissolving in your saliva, balance and equilibrium, these are all special, and the sensory receptors often have multiple cells involved in the process of generating an action potential. So it's more complex. These special senses and their receptors are more complex, and so we refer to them as special senses. So each receptor has a characteristic sensitivity and specificity, right? So specificity is the receptor and what it actually responds to. What type of stimuli does it respond to? So what type does it respond to? That's specificity. Sensitivity is a little bit different. So it's kind of like motor units in a way. Each sensory receptor monitors a given area. So it's showing you touch receptors as your skin, right? So they're called receptor fields. It's this is one afferent sensory receptor. And then that's the axon going to central nervous system, right? And these receptors will branch and monitor a certain given area of the skin. And so if you have someone pushing in, if you have someone pushing in this location, it's going to send an action potential. If you have someone pushing in that location, it'll send an action potential. If you have someone over here. And so the larger the receptor field, the less your brain can be sure of exactly where that stimuli is coming from. If the receptor field is so big, your brain can't really determine, it can only determine a general area of receptor field, but it can't pinpoint it. If the receptor fields are very small, like that, it's going to have a smaller area, right? Because this one sensory neuron is coded for a very small specific area. So being able to localize or pinpoint where sensations are coming from is in a large part due to how big the receptor field is. And that is to say, how many sensory receptors are in that given area. The more sensory receptors in that area, the more you can pinpoint exactly where the sensation is, versus like one sensory receptor to a very large area. You just generally know where that sensation is coming from. So specificity, again, it's due to the structure of the receptor and how it can generate action potentials, and stimulating that receptor is going to be dependent on how that receptor is built and what it responds to. So this is showing you, this is a photoreceptor, only responds to light waves. And specifically light waves in the red, green, and blue spectrum. Very specific. Free nerve endings, on the other hand, and you just have to have any kind of touch on those free nerve endings, and they will generate an action potential. So they're not very specific, like light waves. And then here it goes through receptor fields. So again, small receptor fields make it really easy to pinpoint or localize where the sensory is coming from, or some sensory input is coming from. Interpreting sensory information really depends on your association areas and what your brain has learned, right? And what it's experienced in the past, what sensory sensations have come in, and And so each different pathway is created and built from the time you're born, and what types of receptors have been the most active. The more active receptors are, the more quickly they respond. So that is to say, I'll give you an example of proprioceptors. So the more you use your joints and muscles in a very specific way, and the more you are using them and responding, so say you play tennis, you're using the visual cues and the sensory information coming in to respond with a motor command out. And the more you're using those sensory receptors, the faster they light up or form action potentials, and the faster you can respond back out to your muscles. And so they're quick. If you don't use them, they're a little bit slower, and so you don't react as quick. We just see it as you are, you're coordinated more, you know, the more you play, the better you are. We just say you're playing better. A lot of that has to do with just how quickly you can get your receptors to respond to stimuli and let your brain know so it can have motor output quicker. If you stop using a limb for some reason, say like your foot, and you have some kind of surgery and you're off your foot for a while, you're not using the receptors because your foot is not hitting the ground. You're not responding to different muscle lengths and joint angles and so forth. And so those receptors kind of get a little lazy. And then when you finally get to walk on your foot again, it quote feels weird because your receptors are having to start responding again. Your brain's like, oh yeah, I think I kind of remember this sensation. So it just takes a little while to kind of get things back when you don't use them. So receptors have this ability to activate quicker if they're used more. But some receptors are what we call tonic receptors, and some are what we call phasic receptors. So tonic are always on, like the photoreceptors of the eye, or those monitoring body position, or pain receptors. And then phasic receptors can become activated, but will kind of diminish its response over time. So it'll take more stimuli to generate an action potential after a while than it did initially. So, you know, someone giving you a back rub, you feel it more at first and it diminishes over time. Or like your taste buds, very active in the beginning, and that activity slows down as there's more and more chemicals dissolving in solution. So these receptors can adapt to the stimuli, which means adaptation means it doesn't form as many action potentials even with the same amount of stimuli. So you're adapting to it, and you form less action potentials and you have less stimulation coming into your brain, to your central nervous system. And there's really two types of adaptation. There is sensory adaptation and there's central adaptation. So sensory adaptation is just where the receptor or sensory neurons respond first in the beginning and form action potentials easily, but will become fatigued and form less and less and less action potentials. So phasic receptors, fast adapting, and so they're really intense at first, but they can quickly adapt. And then you have tonic receptors, which are kind of really slow to adapt to the same stimulus. So, so again, going through these receptors up here. I think I had it opposite on that slide. So in different orders, I should say, in a different order. So sensory adaptation is when it just slowly diminishes the sensation over a period of time. That's different than central adaptation. That is where the entire sensory pathway is completely inhibited. And so I could tell you that there is sensory input, but you're not going to be able to detect it in your brain. You're going to think that it doesn't exist. And so the easiest example is dogs, right? So my mom has a dog, and she loves her dog and she spends every minute with her dog. And her dog can smell horrible, but she doesn't smell the dog anymore because she's with the dog every day. So she has this central adaptation, and it's very common with smell. So if you have the same smell, chemical smell, coming up to your olfactory receptors, they're going to stop responding to that chemical. So you literally cannot smell that chemical. That's central adaptation. If you were to get away from that chemical for a while, and then all of a sudden it comes back, then you start to smell it. So you might take your dog to the groomer, dog's super clean, stays clean for a few days. You don't smell that like musty smell, but then after about 3 or 4 days, the musty smell's coming back, especially if the dog's like rolling in dead seal or something. And that chemical, you respond, you go, oh, dog needs a bath. But if you don't take the dog to get a bath and you spend a few more days, you have central adaptation again and you don't smell it. But any new person's going to smell it. Another example of central adaptation is when you're away from your home for a week or two, and you come back and you first walk into your home, it smells weird. So that's what everyone thinks. They're like, oh, my house smells weird. No, your house has always smelled that way. It's just that you had central adaptation to the way your house smelled. Any new person coming to your house, that's what they smell. So it's this interesting thing that your brain kind of decides if there's a sensation coming in over and over and over again, it decides not to respond to it to kind of limit the amount of information coming in. So that's central adaptation. So general senses can kind of be classified based on location. So they can be external, internal, or proprioception. Again, that's muscles, muscle length, tension in tendons, joint angles. That's proprioception. Or it could be classified on the nature of the stimulus that it responds to. So nociceptors respond to tissue damage, right? So mechanical damage, severing the receptor end, burning the receptor end, or having it be freezing cold. They're responding to chemicals. They respond to pain, basically pain. Thermoreceptors are responding to a change in temperature. Mechanoreceptors respond to physical distortion. So if it's your bladder's that big and you keep having urine come in here until your bladder gets this big, the little receptors in the walls of your bladder, and the walls, that transitional epithelium, gets stretched. Stretched is physical distortion. And so they will form an action potential and let you know that your bladder is now bigger. Or pressure, you're pushing on it, right? And physically distorting it. So baroreceptors can be for stretch. So these are receptors that are monitoring the blood pressure in your aorta and carotid sinus, right? If the walls of those areas get too stretched due to too much pressure, it's going to then inform your brain and your kidneys and all the other mechanisms that we have for blood pressure. Chemoreceptors, these are, you find chemoreceptors in your blood as well. So they're measuring the acid in your blood, right? So the concentration of protons. Really that has a lot to do with how much carbon dioxide. They monitor the pH, right? So it can be also the pH or acidity, or which is also this, right? So these two things, so respiratory and pH. Chemoreceptors are also smell, so chemicals, and taste. These are chemoreceptors. So you have to have, for smell and taste, you have to have chemicals dissolve in some kind of solution or solvent, right, to create a solution. And then they can excite a chemoreceptor. So that is to say, you can put sugar on your tongue, but unless you have saliva to dissolve the sugar molecule, which is glucose and fructose, unless you have it dissolved in your saliva, the chemoreceptors for sweet Your taste buds are not going to form an action potential, but if you dissolve it in saliva, it does. All right, so general senses. Pain receptors are nociceptors. These are free nerve endings. They have large receptor fields. So a lot of time pain is hard to localize, right? You kind of generally know where the pain's coming from. There are ones that respond to extreme temperature, mechanical damage, or chemicals. And so all three of those can excite nociceptors. Tactile receptors are mechanoreceptors. They respond to physical distortion or stretch. And again, nociceptors can be different types, right? So sometimes a physician will ask you, is it a burning pain? Is it a stabbing pain? Is it a throbbing pain? They're trying to kind of figure out what's going on. There's also referred pain, and we can get into referred pain and physiology, but it's basically the pain is coming from, say, your heart muscle because you're having a heart attack, but your brain is interpreting that pain is coming from your arm. So remember, it's your brain that's interpreting where the quote pain is coming from. And so that can be fallible. That's what referred pain is. Your brain not quite getting it right. Thermoreceptors, they're found in the skin, in your muscles, in your liver, and your hypothalamus. Your hypothalamus has centers for thermoregulation, and these are responsible for making sure that you keep your stable body temperature, right? That you stay around 98.6. Even when you're exercising, you don't want to go from 98.6 to 102. That would not be good for your body. So you have receptors that if you're exercising and your temperature starts to go up, you start sweating and trying to get rid of that excess heat. So mechanoreceptors, again, touch receptors, baroreceptors, sensing stretch, arteries, bladder, that kind of thing. And then proprioceptors are skeletal muscle length. The Golgi tendon apparati are tension in the tendons as well as joint, where your joint is and what, angle. So this goes through mechanoreceptors and talks about each one of these, and you guys can go through that. I just expect you to understand the basic nociceptors, baroreceptors, mechanoreceptors, those, tactile, that's touch. We went through these. We went through the dermis. And so this kind of goes through all the different mechanoreceptors, baroreceptors. You can stop the lecture and kind of go through these slides on your own because they kind of explain what I've already explained, but in detail, bullet points, and so you could read through those. So let's get to special senses. That's all general senses. Now we're going to talk about special senses. So smell, olfaction is a special sense. And this is cranial nerve number 1, your olfactory nerve, right? And the olfactory system is pretty amazing. It can distinguish between thousands of different chemicals. So when we're normal breathing, we just kind of breathe air, right, this way. If you want to get the chemical to come up to the receptors, you really have to sniff and bring that chemical up to the, basically the cribriform plate where those receptors are dropping through. And then there is this mucosal lining that can bind the chemical and dissolve it. So these receptors will then respond to the chemical dissolved in that mucosal lining. It's a special sense. It has its own cortex where it goes for olfaction, and it has a bunch of support cells. So it's a little more complicated. Interesting facts. So it's a chemoreceptor. I'm really not writing that out very eloquently, but it's chemoreceptor. It's identifying patterns of the different receptors forming action potentials and what pattern did which receptor form an action potential. And sadly, the number of olfactory receptors decline with age. So older people in their 70s or 80s have less ability to smell. Olfactory pathways, so pass through the cribriform foramina, synapses, and that action potential then arrives at the cerebral cortex, hypothalamus, and limbic system. So this is the only sense that does not go or does not synapse in thalamus. It goes more to your emotional brain. We have a lot of physiological responses to smell. In part, it doesn't really get filtered at all. It goes straight to our emotional brain. Gustation or taste, right? We have lots of taste buds. So little kids, right? Kids have 10,000 taste buds, declines rapidly by age 50. Okay, kids, they don't generally like a lot of spices, and they don't generally like a lot of really strong tasting foods like garlic and onions and sriracha, right? A 5-year-old's not begging generally for a burger loaded up with sriracha, onions, and jalapenos or whatever. They have too many taste bud receptors. They have too much sensation. But as people age, the number of receptors decline, and that's where people are just dumping on the spice, right? So you look over, if you've ever, I don't know, noticed, as people age, they tend to want more and more and more chemicals added. So whether it's spicy, whether it's sweet, whether it's salty, just because you have fewer receptors. So you need more chemicals to excite the fewer receptors you have. If you have a lot of receptors, you don't need much chemical to have a response, but as those decrease. So things kind of start to taste not as good because you don't have as many receptors to get excited and bring that quote sensation into your brain. So threshold level, by the way, for gustatory cells for unpleasant is very low. That means that if it's an unpleasant stimuli, it's going to react very easily, right? You're going to form an action potential very easily. But if it's a really good stimuli, it takes a lot more of it to make those particular receptors respond. So we are kind of geared to have more sweet and need a higher amount to excite our taste bud receptors than if it's bad for us or if it's unpleasant. There are really 4, and then there's kind of a 5th and 6th taste sensations. Sweet and salty are in the front of your tongue. Sour and bitter are towards the back, and then umami is in the very back. Umami responds to MSG, or monosodium glutamate. That's why it's called a flavor enhancer. And so flavor enhancers enhance the flavor because they excite more taste buds or more receptors than just the food itself. Right? So if you excite a number of these, right, and you excite a number of these, now you have 2 times more stimulus. And so that's why people use this to stimulate the umami at the same time they're stimulating sweet and salty. So you have a higher perception of stimulation, right, coming into your brain. There's also water receptors. That's why sometimes water can, quote, taste. You can taste water. But this is kind of one of those things that you see in some books, and other books don't even touch on it. So again, this is kind of showing you the umami are back here. And then sweet, salty, bitter, and sour. They respond to different chemicals, right? So sour is going to be an acid, and bitter is going to be probably some kind of protein. Salty is going to be sodium chloride. Sweet is going to be some form of glucose or fructose. These taste buds have these kind of receptor endings here, and then the chemicals dissolve and kind of excite these little areas. And So those are taste buds. They also decline with age. So, you know, people in their 70s, 80s, 90s, things, they're not as excited about food. They can't really smell it and it's hard to taste. So it is kind of a bummer. Okay, hearing. There are three distinct parts of your ear. So the first one is the external ear, and that includes the auricle and the external acoustic meatus, which is the canal. Then you have the middle ear, and then you have the inner ear. So the middle ear is separated from the external ear by the tympanic membrane. We sometimes call that the eardrum. It completely seals off the external auditory canal from the middle ear. And we call it the eardrum because it's actually kind of see-through translucent sheet of connective tissue that acts like a drum. So when sound waves kind of get funneled to your external acoustic meatus, and then they hit the tympanic membrane, and depending on how far apart the waves are and how big or small the waves are, it's going to vibrate. It's going to hit the tympanic membrane like a drum, and that's going to cause mechanical energy to be kind of transferred and intensified by the auditory ossicles. They're the three little bones that take the vibrations of the tympanic membrane and kind of transmit that vibration in each auditory ossicle and increase it, and then send it as mechanical energy into the cochlea. So the cochlea is in the inner ear, and then the semicircular canals and the vestibule are for balance and equilibrium. And this is balance, and really this is equilibrium. And then this is hearing, right? Sound. And that's in the inner ear. The middle ear, by the way, is connected to your mouth, right? So mouth, nose, throat. That we'll go through the narrow nasopharynx when we get to the digestive tract, but it's really your nose, your mouth, your nasopharynx, and then your oropharynx, because they're connected. This is so you have the same, this is so you have the same pressure on both sides of your eardrum. So the nasopharynx you want to have, so you have atmospheric pressure of atmosphere here. You want to have the same pressure on the other side of the tympanic membrane. So there's the same pressure on both sides of the eardrum, or the drum part, and so it will vibrate appropriately with the sound waves. If you go up in altitude, the pressure is lower than atmosphere. So if your pressure drops below atmospheric pressure out here, but it's still atmospheric pressure here, you have to open up your nasopharynx, yawn. You have to open your mouth, bring in the new pressure, right? So bring in that new lower than atmosphere pressure into your middle ear so it can equilibrate or become the same on both sides. That's why if you go up really fast in altitude or down really fast in altitude, you have to pop your ears, you have to open up your mouth and really try to get the new pressure to come into your middle ear. That's why we do that. Okay, so auditory ossicles. You have the malleus, which is the first one. You have the incus, which is the second one, and you have the stapes. Kind of looks like a stirrup, which is the third one. And it is right in front of what is called the oval window. So the oval window is this window into the cochlea. So opening into cochlea. And so we'll talk about that in a minute. So these auditory ossicles are transferring the mechanical energy from the tympanic membrane to the oval window of the cochlea. Now these auditory ossicles are taking the vibration of your tympanic membrane and intensifying that mechanical energy with each auditory ossicle and transferring that energy into the cochlea. Here's the thing, if you have too much energy, then you have these little muscles called the tensor tympani and the stapedius muscle that can contract and kind of hold on to those auditory ossicles to decrease the energy. So that is to say, super loud sounds over a period of time, i.e. a live concert, which is kind of a thing in the past, or you put earbuds in, right? So you have your AirPods in and it's blaring music. That blaring music is loud music. It's going to hit that tympanic membrane with a lot of energy, and then energy is going to increase here, it's going to increase here, it's going to increase there. And you're going to try to protect yourself because if too much energy enters the oval window, that energy is going to crush the little hair receptors in the cochlea. So the tensor tympani and the stapedius muscle will kind of hold those auditory ossicles and try to dampen the sound. That's why after hearing loud sound for a while, if you're not hearing anymore, everything sounds a little muffled and you still hear maybe a ringing or something interesting in there. It's just because those muscles are still active and your perception is a little off from what's coming in. So we'll get to more about cochlea in a second. Backing up here, just one second, I also want to mention something about the auditory canal. It used to be called the eustachian tube, by the way. So the eustachian tube, it's opening to your nasopharynx. Now, when kids are little, here would be their ear, and here's their tympanic membrane. And I'm not an artist. And so anyways, here's the middle ear. And then here would be this would be the auditory tube. When kids are little, this auditory tube actually kind of goes here. And here's the nose, right? So, or the nasopharynx. So when kids are little, their auditory tube or eustachian tube is kind of flat. And so when they get sick and they get a cold and they get a bunch of mucus in their nose because they're not good at blowing their nose, that mucus kind of goes right back into their middle ear. And so little kids tend to get a middle ear infection. As they grow and develop and body parts change, this auditory tube actually goes down at a sharper angle. And so so two things. People learn to blow their nose and not have it sitting in their nasopharynx for long periods of time. So you can get rid of some of that mucus. And the mucus has a harder time going uphill. And so adults don't tend to get middle ear infections because they clear the mucus, and it's an uphill battle with the auditory tube as you age. That happens about 4 years old. So kids get tubes in their ears when they're little, when their eustachian tube is flat, and they happen to get a lot of colds and that mucus just backs up. They put tubes in to kind of drain it. If you have mucus in here, it's really hard for those auditory ossicles to vibrate. And so sound and interpreting sound becomes harder, right? Because you don't have the energy transferring correctly to the cochlea. So that's why little kids that have a lot of ear infections can be speech delayed. They're just hearing delayed. And so they're not getting that sensory speech input. And so they're not programming their brain as quickly. So that's what they're hearing. Okay, so the vestibular complex, which is the semicircular canals and the vestibule, is for balance and equilibrium, and then the cochlea is for hearing. And both of them have a bony structure around them. So this is bone, right? And this is bone. And so you have a bony structure that is called the bony labyrinth. So that is what the bony labyrinth is. And then inside the bony labyrinth, you have a fluid called perilymph, and it's in here. And it's in here. And so perilymph is inside the bony labyrinth, and it surrounds, sorry, I'm getting ahead of myself here, and it surrounds the membranous labyrinth. And the membranous labyrinth really is the blue, right? So this, and this, and this. So that's the membranous labyrinth. And inside the membranous labyrinth, you have endolymph, because endo's inside and peri's around the outside. Both structures. This bony labyrinth with perilymph inside surrounding the membranous labyrinth with endolymph inside. It goes for the cochlea, which is houses the sensory receptors for hearing, and the vestibular complex, which is for balance and equilibrium. And again, the vestibular complex can be broken down into the vestibule, which is the utricle and saccule. This is for linear and gravity, sensing linear acceleration or deceleration, and gravity going up or down. Semicircular canals are for rotational movements, right? And so it's if you're spinning, so you can stand and spin to your left, spin to your right. You can do a somersault forward, you can do a somersault backwards. You can do a cartwheel to your side, right or left side. So each semicircular canal will tell you that you're going a different direction, right? And so that's rotational. They both have the same fluids and hair receptors. They're just a little bit different. So let's look at the cochlea. This picture right here is if we were to cut the cochlea straight down the middle. And then we open it up and we're looking at it. So, so again, this is part of the inside of the bony labyrinth, inside here. And you have perilymph, and this is the other side surrounding the membranous labyrinth. And this would be the membranous labyrinth with the endolymph inside. And so they have names. The scala tympani is this lower one. And then you have the scala media, which is inside the membranous labyrinth. And then above it is going to be the scala vestibuli. And so this goes through those. Now, here's the way it works. Remember I said that the stapes is the last auditory ossicle, and it transfers that mechanical energy into the oval window. And then the oval window, where the mechanical energy starts to move the perilymph inside. And so the fluid is moved by the mechanical energy being transferred into the oval window, and it's like the energy of wind generating waves. So the wind's generating the ocean to move, and it's causing that movement, and it causes a wave. And depending on how windy it is, it's how much of that ocean's moving. So depending on the sound waves, how large they are and how frequent they are, the fluid's going to move a lot, a little, medium. That's all dependent on the amplitude and the frequency of these sound waves coming in, which determines how much energy is transferred, and that determines how much perilymph is moved. If it goes all the way around, it's going all the way around, and there are no receptors for that particular sound wave, then what happens is it gets to this point here called the helicotrema, which is the helicotrema is this point right here in the spiral. And then it starts, the energy just starts to go out the opposite direction. And so then it comes all the way back to the round window and leaves. So the energy leaves the round window if there are no receptors for that frequency of sound wave. That's why we can't hear certain frequencies. We don't have receptors for it. So the energy is still getting kind of absorbed by the tympanic membrane. It's still moving the auditory ossicles. It's still being transferred into the round oval window, but it comes out the round window because you don't have receptors for that frequency. And we talk more about this in physiology, but you do need to understand the cochlea, and you need to understand the parts of the cochlea. And then you have the vestibular cochlear nerve, which is cranial nerve 8. And there's the cochlear branch and the vestibular branch for balance and equilibrium. The receptors themselves are referred to as the organ of Corti, or the spiral organ. This is where you have the little hair receptors that we talk about. These little hair receptors are embedded in a membrane called the tectoral membrane. And when the perilymph goes to the correct place, or how much energy that's generated, that energy is going to push the tectoral membrane, and it's going to bend the little hair receptors, and that's going to start an action potential. And so that's why really loud sounds push harder on that tectoral membrane and can break these little hair receptors if it's repeated over and over and over again. So detecting sound, the sound waves enter the external acoustic meatus. They vibrate the tympanic membrane, which vibrates the auditory ossicles and sends that vibration into the cochlea, and it's going to deflect the tectoral membrane at the location that frequency is coded for. Then the hair receptors will bend and generate an action potential, which is depolarization. So that's hearing. With balance and equilibrium, you have hair receptors as well. They're embedded in kind of this gelatinous material in what is called the cupula. The crista is where the hair receptors are going up, and then you have the sensory nerves. This is a little different. Again, it's in the membranous labyrinth, and each semicircular canal has these, right? So it's showing you each semicircular canal has these, and they're at the beginning of each duct in what is called the ampulla. And when the fluid moves, depending on the endolymph, if the fluid is moving that way and bending the hairs that way, you'll interpret the rotation as this way. If it bends that way, then you're going to interpret the rotation this way. So depending on which way the fluid moves, if the fluid's moving that way or if the fluid's moving that way, we're going to interpret the rotation, left or right, forward or back, to the right side or the left side, sideways. It's going to be dependent. So the lateral semicircular canals, if you were to shake your head no, or if you were to spin around to the right or spin around to the left. The anterior semicircular duct, anterior is for yes, so that would be doing a front somersault or a back somersault. And then the posterior semicircular canal would be like doing a cartwheel to the right or a cartwheel to the left, right? So that's more of a tilting. Vertigo is an inappropriate sense of motion. So you can have these little kind of crystals break off and kind of move these receptors. And you can have this sense of spinning, even though you're not spinning, just because those little hair receptors are being moved. And physical therapists have learned ways to kind of get those little crystals back into place. So semicircular canals, why you feel dizzy after you stop rotating? Well, you have fluid, right? So inside this bony labyrinth, you have fluid moving. And so if you're spinning around and around and around, the fluid's moving. Once you stop, the bone stops, but the fluid still doesn't stop. And so it keeps spinning in those semicircular canals. And so you keep having the sensation that you're spinning, even though you've stopped, just because the fluid keeps hitting those hair receptors and bending them. And that will go until the fluid finally slows down and stops. So that's where you get dizzy. Gravity and linear acceleration, again, you have hair receptors embedded in gel. And on top of that gel, you have crystals called otolith crystals. So you have a gel, hair receptors embedded in the gel, and you have these otolith crystals resting on top of that gel. And when those crystals get separated from the gel, you have a sensation that you're dropping, right? So if you were to drop really fast, this whole thing also drops fast, but the crystals are a different density and they drop at a slower rate. And so they kind of take a while to catch back up. That's why you initially feel the drop when you drop in, like in the rides where they bring you up to really high place. And initially when you start dropping, you feel it more because the gel and the hair receptors in the gel kind of separates from the otolith crystals really quickly. But those otolith crystals eventually come back down onto the gel. If you're going up really fast, it kind of jams into those crystals. So you can feel when you go up and down, just depending on the crystals resting on that gel. With linear acceleration, if you're going back that way, these crystals are going to drag the hair receptors one direction. So you can feel if you're going, if you're stopping really fast or if you're going forward really fast. So that's for gravity and linear acceleration. Pathways for auditory, your vestibular cochlear nerve is coming in. Its synapses here in the midbrain, or the pons, and then the midbrain here, and then the thalamus, and then the temporal lobe. So kind of going through the sensory pathways. Balance and equilibrium really goes to the cerebellum too. We don't get into too much of the tracks and all that. I'm kind of giving you an overview. And then last we have eyes. This is our last sense we're going to go over. And we have external accessory structures of the eye. So palpebrae are eyelids. Conjunctiva. So conjunctiva is going to be the skin that folds. It's a folding under kind of the epithelium or lid. It's where you can kind of pull back your eyelid and you can, flip your eyelids and see the conjunctiva underneath it, right? So that's the epithelial lining of the eyelid. There's the lacrimal gland and the tarsal gland. The lacrimal gland is going to create tears. And so it comes from the lateral side and the tears go this way. And then they'll drain here into the canal that goes into your nasolacrimal duct there. The corners are called the canthus. So you have a lateral canthus and a medial canthus. The lateral punctum, which is actually this thing right here. These are a little off. That's the medial canthus, and that's the lateral canthus. So they're a little bit off, these little lines. And then the external eye muscles. So you can go through, conjunctivitis is when the inside of that conjunctiva is irritated. So it could be due to a virus, could be due to a bacteria. We call it pink eye. Styes, you can have a little cyst on the canthus or one of those areas of the conjunctiva, and so it's like a little bump. So this goes through tears and how they form and go and drain into your nasolacrimal canal. And then we get to the eye itself, and there are 3 layers of the eye, kind of like the ear. So you have the outer fibrous tunic. That is the outermost layer. And then the next layer, middle layer, is the vascular tunic or layer. Tunic really means layer. And then the innermost layer is going to be the retina or neural tunic. So it goes fibrous, then this right here is going to be the vascular. And then the innermost is going to be the neural, right? And that ends it here. what is, the boundary is just called the ora serrata. That is where the retina ends and the pupillary constrictors and dilators begin. And so that is the neural tunic. The fibrous tunic is the sclera, which is the whites of your eyes. And then the cornea, that's the clear lens over iris and pupil. So the cornea actually bends light waves. So light waves come in and then they're going to get bent to go. So one job of the cornea is to bend the light waves to go through the pupil. So the light waves can go through. The sclera is for attachment of extraocular muscles and keep its shape, and so that's what the sclera is. So the mechanical support is sclera, attachment sites for muscles, and then the cornea kind of assists in managing the light waves to the correct location. The next layer is going to be the vascular layer. This is where you have the iris, you have melanocytes that determine eye color. The iris surrounds the pupil. So surrounds the pupil. The pupil is just a hole. It's a hole in which light can go through. So the pupil is just a hole that gets bigger or smaller based on the muscles behind it, these intrinsic muscles that control the diameter of the pupil. And they're on the back side of the iris. There are pupillary constrictors, which make the pupil smaller, and pupillary dilators, which make the pupil larger. That's to let in more light waves or less light waves. So the intrinsic eye muscles on the back of the iris are controlling the diameter of the pupil or hole. There is also in the vascular tunic the ability to control the lens. So the lens is part of the vascular tunic, and you have the ciliary body here, and it's connected with these suspensory ligaments to the lens. And this is musculature, and it completely surrounds the lens and controls the shape of the lens. So that's controlling the shape of the lens. There's also secretion of aqueous humor. This is, the cornea is avascular, so you have to have this aqueous humor here in the anterior chamber to kind of nourish the corneal cells. And if there's too much of this aqueous humor, you get what's called glaucoma. So the lens is also part of this vascular tunic. The neural tunic is the retina, and that is the receptors for light waves, right, and vision. And so this is in the back innermost area, and they have photoreceptors that respond to light waves. There's also supporting cells and then blood vessels. So when you're looking directly, here's your nose, and the light waves or visual access is bringing these light waves in. The cornea, which is here, bends the light waves to go through the pupil, right? And so this right here is the pupil. And then the lens sits behind the pupil, and the lens then bends the light waves even more to then kind of focus the light waves here in what's called the fovea centralis. This is where there's 100% cone photoreceptors for sharp color vision. As you get away from the fovea, we have rods responding that are for dim light, lower light vision. And so rods respond in low light. These are photoreceptors that respond in low light. You can't really see colors with your rods. You can only see shapes and shades of gray. It's the cones that respond to bright light, and they're red, green, or blue cones. And the pattern of excitement of these cones are going to have certain patterns of action potentials, which then when it gets to the visual cortex, you interpret it as colors. And so those cones are in the fovea centralis, and the macula lutea, which is this area surrounding the fovea centralis. The optic disc, which is where all the axons of the ganglion cells of the kind of photoreceptors, where all these axons go and form the optic nerve, there are no receptors. And so the optic disc is called your blind spot because there are no photoreceptors there. Other things, there's an anterior and a posterior chamber that's separated by the iris. And then you have also the anterior cavity, which is the anterior and posterior chamber, and then you have the posterior cavity. Sorry, I said that kind of poorly. Cavity is back here, and then the anterior cavity is split into an anterior and posterior. There's aqueous humor in this area. That's the glaucoma. Okay, so cornea is transparent. You can see through it. It's this collagen fiber layers, and it's continuous with the sclera. This is the fibrous tunic. It's avascular, so it gets its nutrients and oxygen from tears and aqueous humor. You have a lot of free nerve endings in the cornea. That's why you really can feel anything on the cornea, right? Because we want to protect our vision. The iris has blood vessels and pigmented cells, melanocytes. So the more you have, the more brown your eyes look. The less you have, the more blue they look. And then on the backside, you have the pupillary dilators and constrictors. Those are the intrinsic muscles. Those are under the autonomic nervous system control. Whereas your extraocular eye muscles that are attached to the sclera are going to move the eyeball. Those are skeletal muscle. So somatic nervous system. And so this goes through the pupillary dilators and constrictors. Again, this kind of shows posterior and anterior cavity and the different parts there. And again, pupil. I want to bring up the fact that the ciliary body, the iris, the pupillary dilators and constrictors go all the way around the lens. So the ciliary body completely surrounds the lens and are the muscles that changes the shape of the lens. So the ciliary body are muscles, and they're connected to the lens with suspensory ligaments, and they go all the way around the lens, and they can change the shape of the lens to bend light more or less. And so you learn about this in physiology. So for now, you just need to know that's the lens, and that's the pupil, and that's the cornea. But when we get to physiology, whoops, it's important to understand that the lens is changed in shape by the muscles of the ciliary body. So this goes through that area. Suspensory ligaments are what are attached to the lens. And then we get to the retina. So the retina is the photoreceptors for light waves. And this is coming in, so that visual access. So this is the visual access. And so this is the way it's going. So this is light going in, and that's light coming in, right? And so the photoreceptors that are cones, that's a cone, and these are rods. They're back here. You have this pigmented epithelium in the very back. So this is pigmented epithelium. So the pigmented epithelium is this layer. And then the cones and rods are embedded in that pigmented epithelium. And these cones and rods are attached to the next cell, which are called the bipolar cells. And when the cones and rods respond to light waves, they stop inhibiting the bipolar cells. And if they're not inhibiting the bipolar cells, then the bipolar cells can start to form generator potentials. You'll learn about these in physio, but they can start to affect the ganglion cells. And if they affect the ganglion cells enough, the ganglion cells form action potentials. And the axons of the ganglion cells are what form the optic nerve. So the optic nerve is really all of the axons of the ganglion cells. That is to say, this is a special sense because it is more complex. You also have amacrine cells and horizontal cells that are there to kind of support all these different cells, so they're called support cells. But you, again, have this pigmented epithelium with photoreceptors embedded in it, and they are synapsing on the bipolar cells, which synapse on the ganglion cells, and etc. So it's complex. What do you need to know right now? The retina, right? This is the retina. Again, the fovea is where there's 100% cones, whereas your sharpest color vision. Optic disc has 0% cones and rods or photoreceptors, called your blind spot. And the way this works is, again, light waves coming in, cornea bends it to go through the pupil, then the lens bends it to optimally hit the fovea, where there's the high density of cones and rods. So light waves are getting bent through the cornea, through the aqueous solution that's in the anterior and posterior chamber, through the vitreous gel that's here in the posterior cavity, and then will basically hit the photoreceptors. Visual pathway, and by the way, it's hard to see color in the dark because only your only your rods are getting excited. I guess it's the same. You go into Abercrombie, or what is the really dark one? Hollister, you know, back when you went shopping in an actual mall. Hollister can be really dark. Those stores can be really dark. You can't quite tell if it's a pink, an orange, or a red, because the darker it gets, the less color you can perceive. Or at night, you can't tell, you know, which animal that is because you can't see the color of the animal's fur, really. You just see the shape. That's because rods only respond in low light. They don't give you color. So visual pathway. Again, information's crossed from both eyes to both hemispheres at the optic chiasm. So each cerebral hemisphere gets information from both eyeballs. That is so you can piece things together in 3 dimensions visually. Also, if you happen to have one of your eyes not functioning, both hemispheres can still see some of what that one eye is seeing. Your depth perception and your 3D perception is off if you only have one eye working, but both hemispheres can see some of the image that light is bouncing off of. So visual pathways, you know, we're what we call binocular vision. So we have an overlap of both eyes and what light's bouncing off the object into both eyes. If it was panoramic, these are where the animals, say like a horse. I can't even draw a horse, but if I could draw a horse, maybe this is its head and its ears. I don't know. Anyways, horses have eyes on the side of their head. It's looking more like a Star Wars creature. Anyways, one eye can see from here to there, and the other eye can see from there to there. That's called panoramic vision. They don't have great depth perception because they don't have binocular vision or an overlap to piece both sides of the object together in 3 dimensions. So binocular vision is definitely better for depth perception and 3D vision. So what's happening is it's showing you light bouncing off the object is coming in, is coming in, is coming in, is coming in. And so here you have the optic chiasm. Some of it's going there, other of it's going there. Right? And then from here, some of it's going here, whoops, some of it's going here, and some of it's going there. This is the thalamus. So the lateral geniculate nucleus is in the thalamus. So it's synapsing in the thalamus. Also synapses in the hypothalamus and the superior colliculi, which is part of the corpora quadrigemina on the back of the midbrain or mesencephalon. Then it goes back to the visual cortex in the occipital lobes. And obviously color waves, light waves of different color are coming in and being interpreted by both hemispheres as well. So that's where the brain processing comes in. So this is just kind of an overview. We get more into this in physiology, but we have gone through the hypothalamus, the thalamus, the visual cortex, the optic chiasm, the optic nerve. So I want you to kind of review all that and kind of understand. And that concludes the nervous system and chapter 18.